Good morning. We're just waiting for a few more people to log in. All right, good morning again. My name is Nilay Patel. I'm the director of the Bridges to Stem Cell Research program here at CSU Fullerton. I want to start off by thanking CIRM and the people of California to invest in training of our next generation of scientists. As you will hear later on, and as you know firsthand, many of our Bridges alumni have paved the way to change the way the field of regenerative medicine is going to be. It is really an exciting time to be in this field of research. So today we are having our third scientific session. It's on cancer stem cells. And as some of you may already know, the cancer stem cells and the field of stem cell biology are intertwined with teratocarcinoma studies by Gail Martin back in the early 80s. Our first speaker today is Dr. Julie Unternerer, and she has her bachelor's degree from Loma Linda University. From there, she went on to complete a PhD at Yale University in Department of Cell Biology. In Dr. Ira Melman's lab there, she studied how surface MHC class II receptors promote interactions between dendritic cells and T cells. Then she completed a research fellowship with Dr. George Daly at Boston Children's Hospital and Harvard Medical School. And in 2013, she returned back to her alma mater, and she is currently an assistant professor at Loma Linda University in School of Medicine. She has trained 9 PhD students and many more in the training right now. Her topic is going to be targeting ovarian cancer cells to prevent tumor recurrence. Our second speaker is Dr. Mark Chow. He received his bachelor's degree from Case Western Reserve University. He then went on to do an MD PhD at Stanford University. And for the PhD component in the field of cancer biology, he was working with Dr. Irving Wiseman and Dr. Ravindra Majede. And the topic of his PhD dissertation was identification of CD47 as the prognostic predictor and potential therapeutic approaches to identifying human and treating human cancer. And from then, he completed a clinical fellowship in hematology. He was a clinical instructor at Stanford from 2016 to present. During this time, during his clinical fellowship and based on his PhD work, he founded 2 companies. One is called Hepatax, and the other one is 47. The 47 was then acquired by Gilead Sciences, and currently he is the Vice President for Oncology Clinical Research at Gilead Sciences. And the topic of his presentation is therapeutic targeting of CD47, the do not eat me signal in cancer stem cells. And the great thing about our CIRM Bridges programs in some ways are the fact that many of our students from CSU Fullerton and many other programs have actually worked in Dr. Irving Wiseman's lab and are sharing some of the desk spaces that Dr. Mark Chow had worked on. So with that, I will ask Greg to turn on the presentation. Thank you.
Hello, everyone, and I'd like to thank the organizers for the chance to present our work today. I'll be telling you about targeting ovarian cancer stem cells via the Snail Web7 axis. First of all, a brief introduction to cancer stem cells. What are they? Well, there's evidence that they can be normal tissue stem cells that have gone awry, and there's evidence that they can be a transformed cell that's taken on stem cell characteristics. So any cell in the tumor can become a cancer stem cell. Some elements of both are certainly true. I won't be going into which is true today, but I just wanted you to think about those 2 ways of thinking about cancer stem cells. So when we think about ovarian cancer, which is what we are focusing on, the cells of origin are from the fimbriae of the fallopian tube. and they are these basally located cells. That's kind of a true thing about epithelia in general. If we think about this prototypical epithelium, stem cells are in this basal location. How are they defined? Well, they have the ability to self-renew, to produce identical copies of themselves, and they have the ability to differentiate. They can undergo these asymmetric divisions to differentiate into cells that become terminally differentiated in different cell types. If the homeostasis in the stem cell pool is disrupted, they can hyperproliferate and cause hyperplasia, which can be a precursor to tumor formation. So reasons to study stem cells is, of course, for their role in tumor initiation. They also play a role in tumor maintenance and a very important role in tumor recurrence. To me, it's also interesting to study cancer stem cells because of what's called the oncofetal gene expression patterns that are seen in these cells. In cancer, embryonic pathways are aberrantly reactivated. So for example, microRNAs and pluripotency genes can be expressed in a way that's not normal. So I'll be telling you a lot about a microRNA today. So just a very brief reminder that a microRNA is a small, about 22 nucleotide piece of RNA that binds to the 3' untranslated region of mRNAs and prevents translation. So it's an important way to control gene expression. The microRNA LET7 is a conserved microRNA, and it's a regulator of developmental timing. LET7 promotes differentiation and inhibits self-renewal, and it's highly expressed in differentiated cells. I've illustrated that here with the female reproductive tract, and it's negligibly present in pluripotent cells, illustrated here by the inner cell mass from which we get embryonic stem cells. LET7 targets pluripotency factors and oncogenes, and a partial list of its targets is shown here. So several famous stemness or pluripotency factors, including HMGA2 and Lin28 and REX1, are targets of LET7, as are some potent oncogenes, including RAS and c-MYC. And because of this, LET7 functions as a tumor suppressor. As long as it's active in cells, these factors will be suppressed. There are 12 LET7 family members, and it's known that LET7 expression is lost in ovarian and many other cancers, and that low LET7 levels are associated with poor survival. So the situation that I'm introducing you to is that when pluripotency factors like Lin28 predominate in the cell will be pluripotent and able to self-renew. But when LET7 predominates, the cell will remain differentiated. So we're looking for factors that inhibit LET7 because those can explain why cancer stem cells come to be in the first place. And we're doing this by thinking a lot about a process called epithelial mesenchymal transition. As you'll know, epithelial cells exist in an ordered array where their lateral borders are bound to each other by adhesion molecules, and they can receive stimuli that cause them to undergo EMT and express transcription factors such as Snail and slug and twist and ZEB1 that cause the cell to undergo a big change in its biology and become mesenchymal and migratory. Many tumors are of epithelial origin. Carcinomas are epithelial in origin, and they can undergo EMT. Those cells can undergo EMT as well, and that makes the cells more migratory and invasive. So EMT is known to play a role in invasiveness and in stemness. It's known that when a couple of these transcription factors, namely Snail and twist, are overexpressed, that it results in stem cell characteristics in these cells. In somatic cell reprogramming, there's a complex interplay between EMT, which happens early, and MET, the opposite process, which happens later. And it's known that Snail binds to the promoters of LET7, putting it in the right place to regulate it. So we set out to ask whether this also happens in cancer. So Hanmin Wong is the graduate student who took on this project, And one of the things he did was to overexpress Snail in cancer cells. And here we're doing this in a lentiviral expression system. And then as a readout, looking at levels of expression of stemness markers. And we're showing you here Lin28A, NANOG, OCT4, and HMGA2. And shown in black are the control cells expressing empty vector. And in gray are the cells that are overexpressing Snail. And this is normalized to the control cells. And what we see is that when we overexpress Snail, we see a significant upregulation of all of these pluripotency markers. He did this in many cell lines, but I'm showing you here these two ovarian cancer cell lines, OVCAR8 and OVSAHO. He also tested whether when he overexpressed Snail, again shown in gray, we would see a reduction in levels of LET7, and indeed that's what we saw. He tested 4 LET7 family members that were from several of the clusters located throughout the genome on different chromosomes, and we saw significant reductions in LET7 levels when Snail was overexpressed, saying that Snail could lead to this stemness phenotype because of this the effect that it has in repressing LET7 and reducing levels of that microRNA. So he went on to show that indeed Snail directly represses LET7, but I won't be telling you about those studies today. Instead, I'll be telling you about how we went ahead to try to do this in vivo, where we would target Snail and try to reduce the stem cell phenotype of cancer cells. So I'll tell you a lot more about that now. We tested the hypothesis that nanoparticle delivery of RNAi, which is RNA interference using small RNAs to knock down a gene of interest, RNAi targeting Snail will disrupt the cancer stem cell state by restoring LET7 and or depleting Snail, resulting in reversal of chemoresistance, decreased metastasis, and prolonged survival. So today I'll be talking mostly about the tumor burden aspects of this hypothesis. And in diagram form, it's shown here that we want to knock down Snail in order to increase levels of LET7, decrease these stemness factors, and reduce the state that leads to cancer and metastasis and stemness. So what we set out to do is to drug a transcription factor. Now, many of you will know that the druggable targets in cancer are typically cell surface receptors like HER2 and EGF receptor, for example, where since that receptor is on the surface, if we can develop a drug that binds the receptor and possibly has a chemical effect to kill the cell or to inhibit it, that can be used as a therapy. Well, transcription factors are not thought of as druggable because they're hidden away in the nucleus. And any receptor ligand interactions would be difficult because we need to get our therapeutic into the cell. So we developed a nanoparticle approach to do that. And here are some electron micrographs of our nanoparticles. You can see sort of by their texture that they are mesoporous. They're called mesoporous silica nanoparticles, or MSNs. And I'll probably just refer to them as nanoparticles. So we coat the nanoparticles with RNAi, and then those nanoparticles are taken up by cells by endocytosis, and thus the nanoparticles are internalized into the cell, even though we don't have a cell surface receptor to work with. So Hanmin used this approach, the nanoparticle delivery of RNAi targeting Snail, to test this in vitro first in the same 2 cell lines, and we saw the same reduction. We saw the expected reduction in stem cell markers when we did that in these ovarian cancer cell lines. We cared about these studies being clinically relevant, so we wanted to not only test it in cell lines, but also in patient-derived cells, which we did here, and again saw a significant reduction in stemness with nanoparticle delivered Snail targeted RNAi. Next, we checked whether we could see actually a replacement of LET7, increases in levels of LET7 when we knocked down Snail with this nanoparticle approach. And indeed, that's what we saw both in the cell lines and in the patient-derived cells. So this was encouraging that knocking down Snail could reverse the stemness phenotype of these cells. We tested this in several other ways that I won't have time to tell you about today, but encouraged by those results, we went ahead and tried this in vivo. And first we did this in a cell line model where we did microsurgery to inject cells in which Snail was either not knocked down, a control knockdown, or in which Snail was knocked down. And these cells also expressed luciferase, making them visible to us in IBIS imaging. And then we looked over time, these are the days of the experiment, 0, 6, 13, and 20, and saw tumors developing and metastatic tumors also developing. When we measured by IBIS imaging, which can very precisely measure the photons, the result was that the primary tumors were not different from each other. However, the metastatic tumors were highly reduced. There was less metastatic tumor burden, as shown in yellow here, when Snail was knocked down in the cells that went into this experiment. So based on this, we went ahead, and at the end of the experiment, when the mice reached their endpoint and we dissected out the primary tumors and the metastatic tumors, there was a reduction in size of tumors. We move forward then with our patient-derived xenografts, where patients who consent can donate their tissues to us when they have their surgeries to remove their tumors, and then we work with them in vitro to culture the cells long enough to use a lentiviral approach to express luciferase in the cells, and then we expand them in mice, and then we can use them for the experiments. So when Hunman did this, of course, the first question is, are we able to knock down Snail in vivo? So what this involved was twice weekly IV intravenous injections of the nanoparticle loaded with RNAi targeting Snail. And what we observed and were very excited about was that we did get a significant reduction in levels of Snail in the tumors in these mice. We saw in parallel that there was a reduction in expression of several stem cell markers that we've been talking about all along. That was on the RNA level. The same was true on the protein level. And when we tested the tumors for levels of expression of LET7, we saw a significant increase in levels of LET7, meaning that these cells now had increased LET7, which could act as a tumor suppressor. At the endpoints of these experiments, primary tumors were significantly smaller. Metastatic tumors were not in this case. And the IBIS experiments showed that the size of tumor, the tumor burden, was significantly reduced in the mice that received the nanoparticle delivered SI Snail. This was a very encouraging result, showing us that we can, in fact, target a transcription factor and that it can result in an improved phenotype for the mice bearing these human tumors. But this was done in the tumors from one patient. And we, of course, need to do this in more patient-derived xenografts, abbreviated here, PDX. So when we did this with a second PDX, we did see a significant reduction in levels of Snail. We saw a reduction in levels of Lin28 and OCT4 as well. And we saw a very healthy increase in levels of LET7. And there was a significant reduction in tumor burden. Again, in blue are the mice that received the twice weekly doses of nanoparticles loaded with RNAi targeting Snail. But as you can see, in these tumors, the differences were not quite as large as those in the previous PDX. We've thought a lot about this, and what we are aiming for is precision medicine, where we can determine what vulnerabilities specific patients have and choose their therapies based on that. So many studies have shown that there are different subtypes of ovarian cancer. And the first PDX that I showed you, our putative subtype of that tumor is Epi-A, whereas the second PDX that I showed you has the subtype of MES. So it appears to us that we can already begin to delineate that our proposed therapy is more efficacious in tumors that have the Epi-A subtype. Of course, this needs to be tested in many more patient-derived tumors. So in conclusion, I showed you that snail expression correlates with the stem cell phenotype in vitro, and that we can achieve in vivo knockdown of snail via nanoparticle delivery of RNAi. Snail knockdown in vivo results in reduced stemness in tumors and in restoration of LET7 expression and in reduced tumor burden. Because of this, we conclude that snail is a viable therapeutic target in ovarian cancer. And tentatively, we conclude that the epi-A subtype may be more susceptible to snail knockdown than the MES subtype. I'd like to conclude by showing you my lab members two summers ago now. As I've said, this work was spearheaded by Han Min. I'd like to call out my clinical collaborator, Dr. Yevgenia Ayafi, and also my collaborator at City of Hope, Carlotta Galakin, who's here. She did in parallel studies targeting also with MSNs, a different EMT factor called TWIST, where she also got very encouraging results on positive effects on tumor burden. And here's a list of my current and former CIRM Bridges interns. It's been a pleasure to work with all of them. And thank you all for your attention.
Hello, everyone. My name is Mark Chow, and I'm a Vice President in Oncology Clinical Research at Gilead and prior co-founder of 47 Inc. And the title of my talk today for this session is Therapeutic Targeting of CD47, the Do Not Eat Me signal in cancer stem cells. So over the next 15 minutes, what I hope to be able to accomplish and discuss with you all are three things. I think number one is to, again, briefly highlight the concept of cancer stem cells and the rationale for therapeutic targeting in cancer, and specifically liquid tumors. To then discuss, really most of the focus is on the role of CD47, a do not eat me signal in cancer and leukemia, and highlights really a unique bench to bedside story of targeting CD47 in cancer at the start from Stanford University to 47 and now at Gilead, and highlighting how CIRM has played a key role into this. And after this talk, we'll be happy to take any questions. So I think as discussed in this session, there are many cancers now that have been described as being causally related to the notion of cancer stem cells. And acute myeloid leukemia is thought to be derived from abnormal hematopoietic or blood-forming stem cells. And I like to think about hematopoietic stem cells, or HSC, this analogy as kind of the seed of a plant or a tree. It has a unique property to be able to self-renew or create an identical copy of itself, so i.e. another tree, or have the ability to differentiate, which then allows you to create downstream cells or branches that have ability to create the leaves of a tree and furthermore all the components of a blood system. So from one single blood-forming stem cell, you can derive all of the components of a blood and immune system, like macrophages, platelets, blood cells, etc. Now we know that acute myeloid leukemia or aggressive cancer, liquid cancer, can also co-opt this and basically develop mutations to create a leukemia stem cell, which then its only ability is the ability to proliferate all the different leukemia cells that give rise to a cancer. And again, as highlighted here, it's discussed that, again, elimination of this leukemia stem cell or cancer stem cell is really important to achieve long-term remission and cure, because this is the cell type that's thought to be responsible for all the different types of cells or leukemias that arise within that specific cancer type. So back in 2005 or earlier at Stanford, we asked the question of how can we target these leukemia stem cells? And we asked, simply approach was, what proteins or molecules are expressed on leukemia stem cells that are not expressed on normal bone marrow cells? And so when you did this comparison, looking at a bunch of different genes, you found that there were a lot of genes, over 4,000 genes that were differentially expressed, meaning were higher in the leukemia stem cell versus the hematopoietic stem cells. We focused on cell surface antigens because there you can use antibodies to target them. And out of all of those, one of specific marker CD47 arose to the top of the list, in part because of its unique function. And this was the story of how we started targeting a protein called CD47. Now, what is CD47? It is what we call a do not eat me signal, whereby tumor cells actually evade being eaten up by macrophages through signaling of CD47. So this way it serves as a do not eat me signal and allows cancers to evade the immune system. And so I'm going to show you a little video to hopefully show you a little bit more about the details of this concept in this next slide shown here. And so this is a little bit of a cartoon that will walk through the role of the protein and how we can target it. And so as we know, the body is made up of a bunch of, immune system is made up of a bunch of different cells. And their job is to really determine a normal from abnormal cells. And so this is an example of immune cell. This is what we call a macrophage, which we think of as first responder cells, like firefighters or paramedics. Their job is to eliminate and eat up abnormal cells and to basically clear out those that are unhealthy. So when it interacts with a healthy cell, it can detect that the cell is normal, so it doesn't phagocytose or eat them. But when it picks up an abnormal cell or a cancerous cell, its job is to then phagocytose or ingest the cell to eliminate them. And it'll do this and it'll continue to do that, whether it's infected cells, cancer cells, etc. Now we know that cancer cells actually can outsmart macrophages because they can send off a false normal cell and block phagocytosis, and then allow these cancer cells to evade the immune system and grow. How do they do that? So they send off signals called CD47, which again is this false normal sign that tells the macrophage not to eat the cell. So it tricks, the cancer cell tricks the macrophage into thinking that it's normal. So we can take advantage of this process through an antibody that we call 5F9 or MicroRNA that blocks CD47 as a therapeutic. So as you see, these antibodies can now block the CD47 do not eat me signal on cancer cells. And now these macrophages can engage these cancer cells and then figure out that it's a cancer and ingest them. And so this is a way where we can then retrain the immune system to eliminate these to eliminate these cancer cells by blocking CD47. And that's one of the ways in which the mechanism is explained for this this drug that we have developed. Okay, so now I'm going to go into a little bit of preclinical data or lab-based data. And I mentioned that we found CD47 as a leukemia stem cell marker in acute myeloid leukemia, which again is an aggressive white blood cell cancer. You can see in blue, this is expression of CD47, which is overexpressed compared to normal bone marrow cells in red. This overexpression actually leads to a worse prognosis in patients. So we found that when patients have higher levels of CD47 in blue, they actually have a worse prognosis or outcome. And we can further show that blocking CD47 can actually get macrophages to engulf cancer cells. And that's what I'm showing you here in this video. So you can see here right now, red macrophages and green cancer cells with control antibodies. So plenty of interaction, but no actual ingestion. But when we treat with an anti-CD47 antibody on the bottom right, you can now see that these macrophages in red are gobbling up these green cells. And you can see that there are multiple cells being ingested per macrophage. So this is what we think is actually ongoing in the patients themselves. Okay, and so magrolimab is what we call our antibody, which is a novel or first-in-class modulator of macrophages through CD47. All right, a little bit more lab-based data. So we actually took mice and engrafted them with patient leukemia cells and then treated with our CD47 antibody. And what you can see here in blue is actually individual mice on the left treated with CD47 antibody. And even after one dose, you can see that there's essentially near elimination of leukemia in the peripheral blood here. Whereas in red are IgG controls, and you can see the leukemia continues to grow. We know that this targets leukemia stem cells because we can show that there's an elimination of leukemia stem cells based on cell surface markers, and it prevents also further engraftment. And I'll just say to round out the preclinical data, we've shown now that CD47 and the role of this is really involved in many cancers. Almost every cancer we've tested, which has been over 25, now 40 different types. This is an example in acute myeloid leukemia with mice engrafted with human leukemia cells. You can see on the left in blue, all those blue dots are all leukemia in the bone marrow. Whereas you treat with CD47 antibody, you empty that up and that white space essentially has eliminated leukemia. We can show the same thing in solid tumors as well, where this is a picture of a mouse lung. These brown dots are actually human metastases or human tumor infiltration. When you treat with CD47, those brown dots are gone, meaning that CD47 antibody has eliminated those disease. Okay, so pivoting a little bit now to the clinical side, we made these discoveries in about 2009 at Stanford University. So Irv Weissman, Ravi Majeti, myself, and others, and really wanted to develop this into a therapeutic. And so this was actually where our partnership with CIRM started, where we had a collaboration where CIRM was able to provide financial funding for us to be able to develop this antibody within Stanford. This allowed us to start first-in-class, first-in-human studies with the antibody in patients with solid tumors. And I just say that if it wasn't for CIRM, we would not be able to be where we are in bringing this drug to patients. So really a testimony of how this effort really has helped accelerate stem cell research. In 2015, we formed 47 Inc., which is a biotechnology company to accelerate the development of this drug and other drugs in the pipeline. And so I just want to show you a couple of highlights. So actually in 2020, 47 was acquired by Gilead Sciences. And so now magrolimab is being developed within Gilead and has been able to dramatically expand the drug and the program for as many patients as possible. This is an example of data in a clinical trial with magrolimab in a drug called AZA, which is azacitidine, which is a type of drug used in a condition called MDS. MDS is a pre-leukemic condition that affects primarily older people, but has a very poor prognosis where there hasn't been an approved drug in the last 13 years. And in this trial, when you provided the combination of magrolimab and AZA, you saw that 91% of patients actually had an objective response, meaning that their tumor shrank. In over 40%, the disease was completely eliminated. And this shows you what's called a waterfall plot, where you can see that when the waterfall plot is downwards, that is the elimination of the tumor burden. And so you can see many patients actually have significant elimination of their disease. And so this is, while early data, quite encouraging. And we are actually now in late stage registrational studies for this condition. Another condition, so going back to acute myeloid leukemia, the initial indication that we investigated, you can see here that in AML, we see about 63% of patients have a response. Over 40% have a complete remission, meaning that their disease was eliminated. And again, on the waterfall plot, you can see the majority of patients have significant reduction of their leukemia in their bone marrow. So again, encouraging data of which we are now, again, in registrational trials to try to get this to patients and approved. And I do want to go back to the last point. I mean, we initially developed magrolimab as a leukemia stem cell targeting agent, right? So we wanted to ask the question, well, are we actually eliminating leukemia stem cells in the patients themselves? And so we're able to look at the frequency of leukemia stem cells on our clinical trial in patients. And what we found is that, at least in these patients, about 30 patients, you can see that when you started treatment in beige, there was a significant reduction in the amount of leukemia stem cells after starting treatment. And actually, about 40% of what we call putative CD34 positive, CD38 negative leukemia stem cells were eliminating in those patients who responded. So again, more work to be done, but I think initial evidence that we're actually able to eliminate leukemia stem cells, and that has led to encouraging activity and efficacy for patients and improving outcomes. And I do want to end just on 2 slides, but just to highlight here the journey it has been from going from a lab discovery into the clinic and into now hopefully late stage clinical trials that will allow us to bring this drug onto the market. And so in 2009, we published results showing CD47 as a role in do not eat me signal in acute myeloid leukemia. Subsequently, other papers that were published on combinations. We developed this first-in-class antibody, 5F9, or later called magrolimab, within Stanford University, and then started first-in-human studies in 2014 and 2015. We spun out 47 as a biotechnology company, had a couple of partnerships with pharma, additional clinical trials. We actually went public or completed an IPO in 2018, and had further data and encouraging results and regulatory interactions with the Food and Drug Administration. And then in 2020, about a year ago, we were acquired by Gilead Sciences and now continue to develop magrolimab and other assets of the company within Gilead. So quite excited about the future, about what this means for patients. And I think a nice example of how academia, industry, and organizations like CIRM can come together to really accelerate drug development in a novel way. And with that, I'll just end on this slide, just again, giving you a history of some of the people behind everything we've done. I think not only do you need great science, but you really need great people to be able to push things forward, to be dedicated, because as we all know, lab science is not easy, but drug development is also even harder. And so how do you really have that commitment? These are pictures from company folks in the lab. I think at NASDAQ with our IPO, other events we've done, and then our CEO at 47, or Weissman in the middle, one of our founders, and then Dan O'Day, who's the CEO of Gilead in terms of when the acquisition was announced. So with that, thank you so much for listening to this talk. And again, happy to take any questions that come up in the section. Thanks so much.
Great. Thank you for those wonderful presentations. Much appreciated. So we have quite a bit of time for questions and a Q&A. So there are a couple of questions that were already asked, and Julie went ahead and answered it, but I'm still going to ask so that she can share with the audience. So one of the questions that Nitika Parmar asked is, can you try a double whammy, shut down Snail and overexpress LET7?
Thank you. That's a great question. And that's definitely what we would love to do. And we're aiming toward doing that. We first have to do the whole set of experiments where we overexpress LET7 by itself, which is each mouse, each in vivo experiment set is a large undertaking. But then, yeah, we would love to do both. And the other thing that we'd like to do is combine knocking down Snail with knocking down Twist, the 2 EMT transcription factors together. So those are all sort of in the planning stages and getting funding stages, and we hope to do that in the future. Great question.
Yeah, great, thank you. So Lily Chen, Mark Lily Chen is asking, does magrolimab work on other leukemia stem cell types? I know you mentioned that it's present on almost 40 different types of cancer, but are there certain markers that would eliminate when this would be therapeutically available or not?
Yeah, we've looked at several other cancers, and we do find that magrolimab can also eliminate leukemia stem cells in preclinical studies. So some of those that we've looked at have been in solid tumors like bladder cancer, colorectal cancer. When you look at phenotypic cancer stem cells, we do see elimination. So the thinking is that, again, because CD47 is broadly expressed, not only on downstream cancer cells, but also the stem cell population, that we would be able to work on both populations.
Yeah, great. Julie, Uta Grisheimer is asking, Are MES subtypes not cancer stem cell driven?
Yeah, so here's the situation. As you probably know, for breast cancer, there are different types, and each of those types is treated differently. Currently, we're just learning about the subtypes in ovarian cancer. And if you are diagnosed, or I am diagnosed with ovarian cancer, you'll get the same treatment no matter what your subtype. So it's really early days on these. And we don't know, to answer your question, why is it that one would respond better than the other? It's not purely a Snail expression. In other words, you would think that the subtypes that have high Snail expression would respond better to Snail knockdown. It's not that simple because they both have high Snail expression. So we're probing that, and we would love to know, and we still need to test all the other subtypes too, but there must be an answer, but we don't know it yet.
Great. Mark, Mitra Hushman is asking, is it known whether cancer cells will compensate for magrolimab treatment by upregulating other cell surface markers to hide from immune attack?
Yeah, it's a really interesting question. So certainly, I think there is possibility. We've identified CD47 as probably the most dominant do not eat me signal, but we actually have published that there are other do not eat me signals that could also play a role in evading macrophage elimination. So there's two we've published. CD24 is one. LARB1 is also another one, and they seem to be have different roles on different cancers, but the kind of clinical utility of that, I think, is still being explored. For example, those signals could be determinants of response for magrolimab. We're kind of looking at that now. So it's an excellent question. I think there's also, it's kind of what do not eat me signals can cancers use, and then also what opposite prophagocytic or eat me signals do they express, and it's this balance of both that allows us to target them.
Yeah, great. And then Mark, I don't know if you can see Nitika's question. Is there a variation or like anything you want to add based on that? Like, because she's asking about, can they develop resistance to the antibody?
Yeah, I think we don't know yet. What we do know is that CD47 seems to play a central role in many cancers, and there's no reports of actually mutations in the gene CD47. So it suggests that there's a lot of pressure for these cells to utilize the mechanism. So as of right now, we haven't found any mechanisms of resistance like downregulation of CD47 or mutations in the gene, but I think it's still early days in the clinic. And then I think probably there's a follow-up question about CD47 expression in primary versus metastatic tumors or just cancer stem cells. We do see, unclear whether you have higher expression in metastases versus primary sites. We've done a little bit of work there. But again, we think that it's not just the level of CD47, that expression that matters, but also co-expression of these prophagocytic signals. Because we do see some patients that have lower CD47 levels, but still are very susceptible to the antibody. And that's probably because they have high levels of the opposite eat me signals.
Mm-hmm. Yeah. Julie, Dr. Kelly Shepard is asking, how important is it to test potential treatment of primary cancers tissue biopsy versus the cell lines and getting to patient variability?
Oh, thank you for asking that question. I mean, we've learned a lot by studying cell lines, of course, but cell lines are limited in their ability to tell us what's going to work in a patient. A cell line, as we all know, has been living on plastic for decades, and the heterogeneity of cell lines is greatly decreased. So when we work with patient-derived samples, it's much closer to the physiological condition of what the proposed drug would see in vivo in a patient. So we think it's really important to work with materials that are more directly from patients. And apart from that, I should have mentioned, cell lines are variable in their the extent to which they are good models of a disease. For example, in ovarian cancer, we all in the field know that the cell lines that are most frequently used and that have thousands of publications are not well representative of the disease. So I think it's really important to think really long and hard about what your model is and choose a good model to answer your question.
Mm-hmm. Yeah, great, thank you. So Mark, Uta Grisheimer is asking a question about what about CD47 expression in other cell types? Are they expressed, any concerns related to anti-CD47 antibody effects in a non-specific, non-intended way?
Yeah, it's a great question. And I alluded to a little bit of this in the presentation, but yeah, you're exactly correct that CD47 is expressed pretty much everywhere on normal cells. So one of the obvious questions is, well, aren't you going to get massive toxicity by targeting this? The reason why we don't see this, it actually is selective to cancer cells, is because of that second signal needed, that pro-phagocytic or eat me signal. That signal appears to be only expressed on cancer cells and not expressed on normal cells. So when you block CD47 on a normal cell, you don't have a positive stimulus, you don't have an eat me signal, so nothing happens. In contrast, when you block that CD47 on cancer cells, you expose this eat me signal, that positive stimulus, and then that's why you get elimination. So it's this balance of do not eat me signals that lead to our ability to target these cells. The other thing I'll just mention is there's one exception. So red blood cells, as they age, they actually can gain eat me signals. So we do see kind of an on-target elimination of old red cells or anemia that, long story short, we've been able to manage through unique dosing strategies and understanding of the mechanism.
Yeah, great. So Julie, I actually have a question about the nanog expression. I'm assuming that the number of cells, especially in the in vivo studies, that could become cancerous is likely to be very low. So nanog expression is probably in order of like maybe 1 in a million cell, and OCT4 kind of coming on. So how would we be in future, how would we be able to kind of identify those at early stages from a diagnostic and prediction standpoint?
Yeah, that's a great question, and I don't know the answer to.
Yeah.
And really, in the cell lines that we've worked with, we don't see expression of either nanog or OCT4 on the protein level. So we are looking at some of the tissue stem cell markers. We do see expression of Lin28 and HMGA2, so they've been our markers. But getting back to your question that has clinical relevance, could we detect those cells that are there at low frequency and target them specifically? Yeah, we don't know how to do that yet. That would certainly be great, but I don't know the answer to that question yet. What we've been targeting is more a pathway approach, where if we can find the cells that are starting to undergo EMT, those would be the ones that are starting to do a reprogramming like event, right? And so we could prevent that EMT-induced reprogramming that leads to the cancer stem cell phenotype. We haven't directly been targeting the pluripotency factors, and partially because we don't see that they're expressed on the protein level.
Yeah. Great. So, Mark, one of the things that always intrigues a budding entrepreneur is, what are some of the things that the students need to do to see this vision and kind of lay the path for trying to start their own company and, to be able to create a product that is applicable for many, many millions of people?
That's a tough question. I don't know how I can answer. I will try to provide my own perspective. I'll just say this from a personal perspective. I actually thought all along that I was going to stay in academia. And, you asked me whether I was going to go to the industry 10 years ago, I would have said no. I think a lot of it is kind of following opportunities and you have to have a bit of luck, but you also have to have flexibility. For my own personal experience, a lot of it has been just working with other people that have done similar things, and you do all this, you bring drugs to patients. It requires an incredible amount of people. Science makes drugs, but people really are the ones that drive it. So I think it's really one of the epitomes of a team-based approach. I think in terms of kind of opportunities to explore whether going into industry or spinning out companies is what you want to do is, I would just say first, let's talk to people who've done it before, who can provide experience of what they've learned, what they would have done differently. And that's always a good way to understand kind of each person's experiences. And yeah, I mean, I guess if you feel a lot of conviction about something, don't be afraid to follow that. I think within everything we do, science, 95% of experiments fail. And so that's the norm. And so I think it's no different in the clinical side. And so having an ability to kind of stay with convictions, learn from people who've done it before, it would be a starting point of what I would say.
Great, thank you.
Can I chime in there?
Yeah, please, yes.
I know very little about entrepreneurship, but what I have learned is that if you have a unique finding, you need to apply for a patent sooner rather than later, like even before you put it in your poster. So be thinking about this, be talking to your intellectual property office at your university, and at least where I am, they want us to apply for patents sooner and more often. And it doesn't mean they'll all get to this stage of what Mark has done, but at least then you have the intellectual property.
Yeah, I mean, one of our overall goals here is translational research and bringing these therapeutics to clinic. So how we share this information is extremely important. And how do we still allow collaboration? Those kinds of things are very complicated. And hopefully all of us can learn more about that, Mark, we have one more question about, what conditions other than oncology do you see these approaches, the CD47 antibody against CD47 being used for?
Yeah, it's a good question. So we definitely know that CD47 biology extends beyond cancer to actually infectious disease, cardiovascular disease, autoimmune disease, and then also stem cell transplants. There's a number of publications that have come out of Irv Weissman's lab on this, but if you think basically like what diseases may use macrophages as an immune system to escape, you can think about certain things. So from an infectious disease perspective, malaria, hepatitis, those that live in macrophages at life cycle, we're finding that use CD47 orthologs in a similar way. We know that for cardiovascular disease, macrophages actually are a central role in clot formation or plaque formation. So then their modulation of CD47 seems to reduce that. On the stem cell angle, we can think about actually clearing out normal hematopoietic stem cells with CD47 in combination with another antibody to create a novel conditioning regimen for stem cell transplants where you don't involve chemotherapy or radiation, a very targeted approach. So there's a lot of different possibilities. I think we're excited. There's obviously too many to potentially translate at one time, but I think for the field in general, it'll be really an exciting time to see where this goes.
Yeah, some wonderful, unexpected but positive side effects. So that's great. Yeah, great. Well, thank you very much for both of you for the wonderful presentation. And I think that we will break for lunch a little early. So if there are no more questions, thank you.
Thank you.
Thanks, everyone. Good session.
Bye.